With that, then, we want to move on to the next session in this public part of our discussion, and that has to do with broad considerations for the science and potential societal applications of the gene drives research. We have a series of investigators here, 5 individuals, who are going to be making presentations for us. Each has been invited to give a 15-minute presentation. We'll let you know with 5 minutes to go that you're nearing the end of your time. All have promised to stay within the 15 minutes of time. Then at the end, we'll bring them up and have another series of Q&A from them at the end. So with that, let me start with Dr. Kevin Esvelt from Harvard University, and he is going to talk to us about gene editing and gene drives. Kevin?
All right, thank you, Jim. So I've been involved in the development of CRISPR genome editing technology, and especially RNA-guided gene drives from early on. And so I'm going to give a general overview of the state of the field and where we need to go from here in terms of the scientific perspective. If I might ask, where's the slide advancer? Oh, here we go. All right, so first I'd like to emphasize that natural selection really does limit ecological change. That is to say, when we alter an organism, we almost always reduce its ability to survive and reproduce in its ancestral habitat. And that means that in general, engineered traits seldom, if ever, spread in the wild. Now, gene drives are exceptions to this rule because they are natural genetic elements that distort inheritance in their favor. So whereas normal inheritance, genes have a 50% chance of being inherited by offspring. In distorted inheritance, a gene drive element can ensure that it is inherited by up to 100% of offspring. So this allows it to spread through populations, even if it reduces the fitness of an organism. And so this is why, by harnessing gene drive elements, we could potentially alter the traits of wild populations. Austin Burt was not the first to propose the use of gene drives, but the first to propose the use of endonuclease gene drives specifically, which gave rise to the project that Phil and Stephanie were talking about. I should distinguish, first of all, that there are two different kinds of gene drives, broadly speaking. There are threshold-dependent gene drives. So these are ones that, if they start out at a low frequency in the population, they will actually vanish over time. And only if they start out at a high frequency in the population, above the threshold, will they spread to take over the population. The other kind is threshold-independent gene drives, where in principle a single organism released into the wild could spread that trait through the entire wild population. So I'm going to restrict my remarks to these. So first, why are we here? It's because of the development of CRISPR-Cas9 genome editing technology. And the way we and other labs originally developed it, we delivered into the cell separate pieces of DNA encoding Cas9, which is molecular scissors that cut DNA, guide RNA that tells it where to cut, and an altered version of the gene we want to edit, such that Cas9 and the guide RNA together cut the target gene, resulting in the cell inserting the altered version in its place. If this is done in a germline cell that then gives rise to an organism, the organism is transgenic for the altered gene. This has been done for a tremendous variety of organisms. But again, the vast majority of Cas9 genome editing experiments pose no risk whatsoever of altering wild populations because, again, the change reduces the fitness of the organism. What is different is when you, instead of delivering those pieces of DNA separately, you encode them all together, such that you incorporate the editing machinery next to the change you're making. So now you have Cas9 and the guide RNA, again, cut the target gene that you want to edit, and the altered version gets inserted along with the editing machinery. Now the cell encodes that editing machinery, which means it's almost certainly going to cut the other allele, and consequently you're going to end up with 2 copies rather than just 1. What's really important, however, is when this organism mates with a wild-type organism, because all of the offspring, since there were 2 copies initially, are guaranteed to inherit 1 copy, and the other copy will be wild-type from the wild-type parent. But now they encode the editing machinery, and so Cas9 is going to cut the wild-type copy, and editing is going to happen again, converting this heterozygote organisms to homozygotes, and thereby ensuring that all of their offspring, when they mate with wild-type organisms, will again inherit the altered version. And this occurs on and on down through the generations until conceivably every organism in the population has the altered trait. Now, RNA-guided gene drives, this form of editing, could spread most Cas9-based alterations through the population. And potentially a single released organism is all that is required. It's important to note that, I'm not going to go into this in detail, but Cas9-based gene drives could be evolutionarily stable in ways that was very difficult to achieve with earlier homing endonuclease technology, or really any other technology that does not allow you to cut multiple sites. And it is not theoretical. To date, it has been demonstrated in practice in both yeast and fruit flies. In the yeast, the efficiency of copying in all cases was over 99.5%. This was done in a variety of yeast strains from all over the world, didn't appreciably affect it. And we also tested many different kinds of gene drives targeting different types of genes, and it worked very well for all of them. In fruit flies, the only extant example is the one case, which was one strain of fruit flies at one gene, but again, the efficiency of copying was 97%. And of course, this is a more relevant model organism than yeast are. Limitations, equally important. So first, sexually reproducing organisms only. It does not work in bacteria, viruses, or anything like that. And there is an argument that it will not work as well in organisms that can reproduce asexually as well as sexually. Fast reproducing organisms only, unless you are willing to wait a very long time for the change to take effect. If you can only release one drive organism per 10,000 wild type, then you're looking at least 16 generations before it spreads to the entire wild population. The difficulty of building a gene drive is going to vary between species. That's because copying the gene drive is not the only repair pathway that the cell can use. It can use other pathways, just jam the ends together. And the frequency with which it chooses one pathway versus the other varies between organisms, between cell types, between developmental stages. So just because the two extant examples have very high frequency, previous examples with standard homing endonucleases showed lower frequency even in fruit flies. So we don't know. 4, you have to be able to make a transgenic organism with Cas9. If you can do that, the odds are that you can build at least a basic RNA-guided gene drive. It is the barrier of making a transgenic sexually reproducing organism that is currently limiting when it comes to making gene drives. That said, there are now high schools that are incorporating Cas9-based genome editing in their curricula. So to give you an idea of where we are heading with this. Not yet in Drosophila usually, but it's only a matter of time. Capabilities. So this is extremely important. Reversibility. The edits that are spread by an RNA-guided gene drive can be reversed or blocked by other subsequent gene drives. So that is the change that you are spreading, the altered version of the gene. However, once a gene drive has spread through a population, you cannot get rid of the Cas9 gene and guide RNAs. Now, it's unlikely for most organisms that they're going to do anything. We have made several different transgenic organisms that express Cas9 and guide RNAs all the time and show minimal phenotypic effects, but you don't know. It's all equally important to realize that even if you can undo the genome change that you've made by the drive, that doesn't necessarily mean that you can undo any ecological changes that may have resulted in the meantime. But I should note that we have demonstrated this reversal effect in yeast. That is, we did spread a change through yeast with a gene drive and then build a reversal drive that undid that particular change and restored the gene that we had broken. The second major capability of gene drives is population suppression, which is, again, what Phil and Stephanie were referring to. That is, there's two different ways of doing this, both of them first outlined by Austin Bird. You can drive deleterious changes to build up genetic load in a population, causing it to crash. Or you can bias populations towards one sex. Obviously, a population of all males will eventually die out. It's important to recognize that suppression drive systems are more difficult to build than standard gene drives because you have to tightly control when the Cas9 or other nuclease is expressed. Finally, there's a lot of more complex alterations that one could imagine making. One might make a population uniquely vulnerable to an otherwise harmless compound to be a species-specific pesticide that doesn't harm any other living thing. You can imagine there are ways of splitting a population into reproductively incompatible groups, instant speciation. You could recode a population to make it uniquely susceptible to a future gene drive for more precise targeting. There's lots of different things that you could do, many of which we outlined in our original publication. Applications, I'm just going to skip over these briefly because we've already talked about them. Obviously, control of vector-borne disease in particular is a major one in health. Sustainable agriculture. We might block harmful pest behaviors such as swarming, in, for example, desert locusts. We might sensitize pests and weeds to, again, unique pesticides that are harmless to everything else. We might even eventually learn how to reprogram olfaction to discourage crop consumption by these pests. Or in the case of mosquitoes, we might make them dislike the smell of us. Finally, ecological conservation. We could control or locally eradicate invasive species, potentially restoring damaged ecosystems. We might also spread protective genes through threatened populations. I want to address the potential for misuse very briefly. First of all, it's minimal, and the reasons is primarily that this is readily detected by sequencing. That is, if you sequence an organism or even environmental DNA where that organism is present, and there is a gene drive in there, you will see it. It effectively cannot be hidden at all. A lot of researchers have been working on hiding genetic changes. As yet, no luck. So you will see it. Once you've seen it, you know how to build a reversal drive that can undo it. You cannot effectively target humans because our generation time is too long, and also future generations could undo it. You cannot effectively impact crops, at least in the developed world, because, again, the seed farms are tightly controlled in their genetics. And suppression drives, which are probably the most damaging kind when it comes to ecological change and deliberate misuse, are much harder to build than standard drives. On the other hand, it is widely and increasingly available at very low cost in terms of parts you could buy on eBay and assemble it, probably less than $10,000 you could do it. And the ecological impact, again, as Phil echoed it, if there is any, is going to depend entirely on the species and alteration in question. So what do we need to do from the research perspective? We need to understand the evolutionary dynamics of these. We need basic research. We need to understand effects on fitness, the stability of the construct, its evolvability, and its potential for spread into related species. We need to do this in model organisms with very, very large populations to mimic the wild conditions as effectively as possible. We need applied research in terms of developing the actual candidates for release in all these different organisms. And eventually we will need field trials, and ideally first determining ecological impact without actually releasing a drive-containing construct, or at least not a functional drive-containing construct. So how can we pursue these safely and responsibly? Well, first there are extrinsic confinement methods. So these are ones that can fail due to human error or deliberate action. First, there are barrier methods that prevent organisms from escaping the laboratory. Then there's also ecological confinement, and this ensures that escaped organisms are not going to find mates or persist in the environment if they do get out of the laboratory. So again, working with Anopheles gambiae mosquitoes in Boston or London would be an example of this. Then there are intrinsic confinement methods, where the probability of failure is entirely unaffected by what humans do. So there's reproductive confinement, work in an organism background that can't reproduce with wild populations. There are several Drosophila lines that are like this. And molecular confinement, build drive systems that only function in laboratory populations and don't function at all in wild populations. So you can split the components. So say you can build a gene drive cassette, I'm putting that in quotes because it would contain only the guide RNA, but that would still function as a drive in organisms that supply the Cas9 component. But it wouldn't work once you went in the wild because they wouldn't have the Cas9 present. Alternatively, you can build a complete drive system, but you can program it to target a site that is absent from wild populations, but is present in your laboratory populations. This is probably the safest form of confinement. I would like to note that in some of the history of this, we mentioned the Drosophila study. The fruit fly work was independently conceived and developed as a genome editing technique and not as a gene drive. So it's actually a great way to quickly make homozygous transgenic organisms, in this case fruit flies. And the authors used a very stringent IBC-approved barrier protocol to prevent escape. But the IBC was unaware of the intrinsic molecular safeguards that were possible, such as, in particular, the split drive, which could have been used for this work and to achieve the same results. And the work was published without either the editors or the peer reviewers flagging this. And that set a concerning precedent since, again, the same results could have been obtained more safely. So again, this is a case of the field just advancing very, very rapidly and science being a very large area, especially CRISPR. So we have since joined with the authors of that work, and we jointly convened a diverse group of 27 scientists, entirely laboratory scientists, to devise consensus recommendations. And this is largely to provide guidance while this panel comes out with its formal guidelines. And those are published today in Science. And I think this is a demonstration that scientists, for our part, we can approach powerful technologies with caution and reach consensus. But only awareness, visibility and a culture of responsibility can ultimately prevent accidents and loss of trust. So my take-home points are, again, this is a tremendously promising technology. The fact that it's reversible, at least to some extent, is tremendously important. And while there are fewer opportunities for misuse, it will be widely available. There is no societal precedent whatsoever for a widely accessible and inexpensive technology capable of altering the shared environment, and therefore lots of people. None. We have not dealt with this before. Specific recommendations. In the area of basic research, because you don't need to alter a wild population at all, you can use the intrinsic molecular and/or reproductive confinement methods. And we recommend that all laboratories do use those and at least one other strategy. In terms of applied research, you can't use the intrinsic methods because the goal is to create a gene drive that will eventually spread in the wild. So you are limited to barrier and ecological confinement. But you can also, at the same time as you build your candidate for release, you can also build a reversal drive that undoes whatever change you're planning to spread, which would also be prudent, and I would recommend that laboratories do that. Any exceptions to these rules and guidelines, in my opinion, should require great scrutiny and explicit higher-level authorization than the standard process. It's not to say that there will not be required exceptions. Some organisms we might want to use gene drives in particular for the applied research, are endemic throughout the world. You cannot use ecological confinement. And so if you can't use those, you're limited to barrier only. And in many cases, this will be worth it. We will need to do this, but it should require greater scrutiny to make sure that no mistakes are made. Finally, I would like to note that the principle of informed consent suggests that all gene drive research should be fully transparent, ideally from the project proposal stage onwards. This is really a unique field in terms of its accessibility and its potential to impact large populations of people from around the world. And I think that if we're going to do this in the safest possible manner and also the most ethical possible manner, personally, I would argue that transparency is our greatest weapon, because again, without that visibility, without awareness that this is a problem, without establishing cultural norms of being cautious around this sort of technology, then we're going to have trouble given how widely available it's going to be. I've set up a comprehensive resource with a lot of different links to save you time, so you can just go to this one site. And then cited the major papers are at the bottom, with stars being the most relevant ones. Please utilize those for your future deliberations. And while I have a lot of people to thank, I think it's important to note that this is not so much about people. This is about everything else. Thank you very much.